I will invite Miss Yagut Allahverdiyeva to the stage to give her speech, but before start her speech, I would like to share a few notes about her. Professor Dr. Yagut Allahverdiyeva-Rinne from University of Turku obtained a PhD degree in Azerbaijan. During her PhD studies, she was awarded with the one of UNESCO fellowship to visit Zent İsfahan University, Budapest. She has joined the molecular plant biology group at the University of Turkey, Finland, as the postdoctoral research fellow in 2002. She has been acting as principal investigator of the photomicrobes group at the University of Turkey since 2010, and currently she is a full professor of basic and applied photosynthesis. Moreover, she is chairing the Nordic Center of Excellence North Aqua, funded by Northforsk Bioeconomy program and focusing on algal biotechnology. She is a director of Biocity Turku research program, Smart Bio, and she is involved in several European, Nordic, and national projects as a partner and coordinator. She is coming to the stage with the speech title of Algae and Cyanobacteria as promising but biocatalysis for direct solar chemicals production. The floor is yours. Thank you.
Thank you very much for introduction. And I would like to thank Berat and the organizer for inviting us, giving us opportunity to talk about our research. But I would like to start from some words about condolences. Depremde hayatını kaybedenlere derin hüzünle başsağlığı veririk. Sizin acınız bizim acımızdı. Bir neçə gündür Azərbaycan yatmır. Sizinle birlikte bu felakete galip gelmeye istiyor. Sözümüz yok ama siz tek değilsiniz. Algae and Cyanobacteria, promising biocatalysts for direct solar chemicals production. So I would like to tell something about what we are doing at University of Turku in Finland. So algae, Cyanobacteria, we call it microalgae. Now they are promising sustainable production platform. This is already known. You have heard about it a lot. I didn't want to have very strong. So they can use also, there is no laser, no. Okay, they can use, okay, I got it, yes. They can use wastewater, water, or they can use flue gas, CO2 to make a biomass, and that biomass through biorefinery can be converted to biofuels, chemicals, and different food and feed applications. And this is, we call in our lab, it's a short-term application strategy, because this can be done in near future and it can be industrially relevant production platform. We are working on it, and tomorrow you will hear from Sama what kind of strategies we have in this part. But we are mostly considering also different route, which means that we are considering to use photosynthetic cells as biocatalyst. The reason is that biomass is not enough. With the growing population, we are getting and more demand on biomass. The EU recently identified that in 2050, about 40–70% gap can be biomass can be in 2050. Biomass gap can be about 40–70% because the increase for biomass demand is increasing, but supply cannot manage to give all the biomass which is needed for bioeconomy, growing bioeconomy. And that's the reason that we need new technologies which can mimic nature, do the same like nature is doing, however it can be much more efficient. One possibility is that to create synthetic cell factories where photosynthesis can be used to uptake CO2 and nitrogen and to produce certain targeted chemicals, and that chemicals are secreted out and milked out, which means that in this case we don't need to grow more biomass and more product, but rather to use the cells as a catalyst to convert solar energy CO2 into certain product which will be secreted out to the media, and you can milk it. Like this, the system will work much longer. So this is the photosynthetic electron transport. I will not go into the details, but I would like to mention that all sustainability starts from photosynthesis. This is the most unique process when sunlight is captured by certain photosynthetic pigment protein complexes, and water is splitted under natural condition, physiological condition, and electrons are then reduced to NADPH, and CO2 fixation occurs. So this process is the unique process because there is a water splitting under natural conditions, and this is this process when carbon fixation occurs and the high value carbon compounds are produced, they use the solar energy. What we can do, we can try to improve photosynthesis. We can make more solar to product conversion efficiency higher, and that we can make a high efficiency by direct solar chemicals production. And in this system, we are studying in my lab how this system is regulated and the different components, what's the role of those components in photosynthesis, regulation of photosynthesis. But now I would like to talk to you shortly about the hydrogenase. You can see here that light is captured by photosynthetic pigment protein complexes, and then water is splitted, electrons go through these cofactors and reduce Na to NADPH, and this is used for CO2 fixation. However, the naturally Chlamydomonas has another enzyme which is called hydrogenase, and this hydrogenase enzyme is able to capture the electrons from photosynthesis and produce hydrogen. So it can be direct water photolysis in natural condition. This has been of interest already many years, already 1940. You can see here, Gaffron and Rubin, they did this kind of experiments. They made the anaerobic condition. I have to emphasize that hydrogenase is oxygen sensitive. It needs anaerobic condition. And when they shine the light, there is a hydrogen production, burst of the hydrogen production, very strong, direct water photolysis. And however, this doesn't last longer because photosynthesis also splits water and produces the oxygen, and when oxygen starts accumulated, hydrogen production stops. And there is several steps how to reach this one. You have to spin down the cells, change the medium, and so on. And then in 2000, it was again big boom that there is a new protocol which can give us possibility to have a sustainable hydrogen production, and that came from USA, from Melis group. What they did, they nutrient deprived the cells. So during the nutrient deprivation, one of the photosystems starts to go down, becomes inhibited, and then during that time, oxygen evolution decreases and the respiration takes more oxygen, and after about 24 hours, you can see that the cells in photobioreactor become anaerobic and immediately hydrogen starts to be produced. And this is very nice protocol. It was big hope there, but the problem here is that cells are not fit. They can do this process for several days, maximum 1 week, because they are nutrient deprived. And also the system is very sensitive. The trace amount of the sulfur, for example, can change the system. And that's why it did not go to industrial application neither. However, it was quite big interest, and still there is a big interest to this production system. Considering these bottlenecks, we decided to make to use our knowledge in photosynthesis in electron transport and to make a new protocol for hydrogen production. You can see here, this is the linear photosynthetic electron transport, water splitted, electrons end up here, and it can end up in hydrogenase, which would mean that water splitting and hydrogen production. However, this is the very sensitive production enzyme to oxygen. And at the same time, there is a competition for electrons at this level by hydrogenase enzyme and CO2 fixation, and that's why we have to consider in our system what kind of system we can make that there is no so much oxygen, but at the same time photosynthesis works well to contribute electrons to the system, and there is no competition with CO2 fixation. So what we did, we made a new pulse method. In this pulse method, without any changing medium, you can take directly chlamydomonas and start applying pulses. And that pulses should be light pulses, very short, because during the short light pulses, CO2 fixation is not active, it's inhibited. But photosystem 2 is working and splits the water. And during these short pulses in dark background or low light background, hydrogen can be produced. We can see here that when you bring the cells directly from the Erlenmeyer, of course it should be gas-tight photobioreactor, and start pulsing, oxygen level goes down because photosynthesis consumes quite much oxygen in the system, and very soon cultures reach the anaerobios and hydrogen production starts to accumulate. And in this system there is no deprivation and there is no any extra steps, so it is much easier to upscale. And what we see that the point why there is a continuous hydrogen production is that CO2 fixation is inactive. You can see also here, if you make a longer light pulses, you start activate the CO2 fixation and there is no hydrogen production anymore and there is an accumulation of the oxygen. As a next step, we were thinking, okay, here, as a concept, what I was talking about, the concept of the biocatalyst, you can see that indeed here cells act as a biocatalyst. This is the starting point. When you have an OD, you can see the color of the culture. And this is the hydrogen production time with the pulses. Color doesn't change because cells do not accumulate biomass. Culture doesn't accumulate the biomass, but instead it produces hydrogen. All solar energy is directed to hydrogen. But in this case, when you apply continuous light, CO2 fixation is working and cells start to accumulate biomass, culture starts to accumulate biomass, and all solar energy goes to biomass rather than hydrogen production. And I jump here, and we further used our knowledge on photosynthesis because we know that there is a flavodiiron proteins which can take electrons directly from electron transport chain and give it to oxygen, produce the water. And this can be waste of the electrons under certain conditions, but it's important for the cells to survive. And when we delete with genetic engineering this protein, what we could see is that hydrogen production is much higher, which means that more electrons are directed to hydrogen rather than to flavodiiron proteins. And in the next step, we did also recovery phases for 4 days about, you have a hydrogen production continuously. You can see here, red is the mutant, flavodiiron mutants. And after 4 days, we give them 24 hours continuous light to let them to recover. And after that, again, hydrogen production and again, hydrogen production. And you can see that you can go over quite long time, about 1 month, even more, to produce continuously hydrogen production with fit, healthy cultures. And we have another, for example, also application strategy to use cells as a biocatalyst. Instead of hydrogen production, for example, we can also heterologously overexpress certain oxidoreductases. For example, in this case, N-reductase can be, CHMO, can be overexpressed in the cells, and that enzymes can take directly photosynthetic NADPH and convert the exogenously added substrate to product. This can be done also in coli. There is a lot of work on E. coli, but E. coli is heterotrophic organism. You need to give them, to feed them with sugar, which means that they compete with the agriculture. But in this system, what we offer NADPH coming from sunlight, which is available there all the time. And you can see here that we did two-step process. When we start anaerobic conditions, in that case, hydrogenase is active because they are active when oxygen is not there. And you can see that there is a hydrogen production, the red line. And after, for example, a couple of hours, when oxygen started to be accumulated, then hydrogenase become inactive, and the heterologously expressed enzyme starts to be active, and it starts to convert cyclohexanone to caprolactone, which is also polymer of the plastic polymer. So this is the one proof of concept how you can use the cells as a biocatalyst for production of the hydrogen simultaneously for production of the caprolactone, which could be precursor for the plastic. The similar system we managed to also do in cyanobacteria, and in this case we used N-reductase enzyme, you can see here, and this is the N-reductase, how it converts the certain chemicals what you add to targeted chemicals, and again this can be done in industrial level, but in this case we offer physiological conditions and environmentally friendly environment where you can produce the targeted chemicals. And again, by deleting flavodiiron proteins, which can be still here competing with the heterologously expressed enzyme, we are able to produce much more product compared to wild type strain, which doesn't have a flavodiiron protein deleted. And on terms of the photosynthesis, what we realize that there is a many alternative electron transport chain players. If we have a very strong enzyme which can take as much as possible electrons from photosynthetic electron transport chain, then that enzyme will outcompete with the alternative electron transport chain, which is important to understand how photosynthesis is regulated and how to improve biotechnological applications. You can see here that in case of inactive enzyme, if this heterologously expressed enzyme is inactive, 17% cyclic electron transport occurs in the cells. But if we add a substrate, then only tiny amount of the cyclic electron transport occurs in the cells. So by understanding photosynthesis, we can further improve the system and to get more targeted products. Another strategy to use cells as a biocatalyst is immobilization. So what we do, we immobilize the cells in certain polymer matrices, and the main idea is that not to divide and to give them safety compound, let's say scaffold. This is a two-stage process. In first stage, you have to have a big amount of the biomass. In the second stage, you immobilize cells in thin layer of films. We call it artificial films or artificial leaf. And then that leaf will produce certain targeted chemicals which will be milked out from the system. So there is quite many advantage of using solid-state cell factories or immobilized cell factories. The advantage is that if you have these suspension cultures are very diluted usually, and this is the big problem. You have to mix them all the time. You put a lot of energy for mixing, for harvesting product, or changing media. Again, you have to harvest the cultures and change the media. And also short catalytic time, because you grow the cells, cultures, about 2-3 weeks, 1 month, and you have a certain amount of the biomass. You have to often give some nutrient deprivation that they produce certain compounds, and it takes 1 week and then cells are dead, all culture is gone. But in this case, if we go solid-state cell factories where we make artificial leaf, we can overcome those bottlenecks. For example, in this case, cells can live in this artificial film much longer. They are able to live there half a year and doing the converting solar energy into targeted chemicals and secreting out, which means that we strongly increase the lifetime of the cells, catalytic time of the cells. And also, most importantly, volumetric production is much higher, because here you can go maximum 1-2 gram per liter, 5-6 gram per liter cells. But in this case, you can go up to 10, up to 20, up to 100, so it's like a paste. And this gives also you next possibility to produce much efficient and much more targeted solar chemicals. And we use different matrices, and we are also collaborating with the material science, and also currently we're starting 3D printing artificial leaves. So for this one, we need to have a matrix. What we need porosity, because should be there exchange between medium and gas exchange between cells and medium. And for that one, the matrix scaffold should be quite porous, and it should be transparent, because we are working with photosynthetic cells, and it should be mechanically strong, because you are using it still inside of the medium, and also it should be tunable, because if you have a specific wheel, for example, you don't want oxygen to go inside of the matrix, then you should be able to tune the matrix to do that one. So that's why we are using also nanocellulose, tempo-oxidized nanocellulose, which is tunable matrix. And what we did, for example, we engineered the cells, we add the Fe enzyme from Pseudomonas to Cyanobacteria, and Cyanobacteria starts to produce ethylene. And usually, you can see here, when you maximize the production system, we can go maximum 2 weeks of production, then cells again culture dies. But when we immobilize them in thin layer films, made the artificial films, we can go over several months. You can see here 3 months production, and this is the just ongoing research, so we will optimize it further. And you can see now we have a continuous production where we have here thin layer film, and then here we can change the parameters of to make much longer production system. And another thing, for example, an example is the filamentous cyanobacteria. Filamentous cyanobacteria anabena, they have a heterocyst, which we call it nitrogen-fixing microoxic factories. Here, there is no photosystem 2, that's why there is no oxygen production. So inside there is a nitrogenase, which can fix the atmospheric nitrogen to ammonia. So these are the nitrogen-fixing factories by cyanobacteria. And what if you isolate that nitrogen-fixing cell from filaments, they can live under atmospheric conditions few hours maximum. But when we immobilize them, we are able to run this system again 1 month, and now we are trying to go over a month, what means that you have an immobilized thin layer of film which can use atmospheric nitrogen and fix that nitrogen and to make ammonia or as a byproduct hydrogen, depending what you wish to do. And as an example, we are also coupling different systems. For example, you can have a biotransformation by E. coli. As I mentioned, in this case, the system is not sustainable because you need a sugar, but we have a sucrose-producing cyanobacteria which can make a sugar and secrete it out to the medium. And we usually, what we did, we immobilized them in small alginate beads, and they secrete out sugar, and that sugar we collect and we give to E. coli to produce certain product. In this case, it's a caprolactone again, which means that we couple this system and try to make a system more sustainable. So our aim is to mimic leaf structure, leaf anatomy, and to make a tailored artificial leaf which can take solar energy, which can take CO2 or nitrogen and produce targeted chemicals. So we have now ongoing European project here, and here is I'm 3 minutes late. Here is my group from our Pikku joulu, which is traditional Finnish Christmas party. And these are the people who are involved in this particular research, which I mentioned now. And we have a lot ongoing different collaborations with European scientists, and we are trying also to build something with Berat. But these are the people who are directly involved in the slides, what I show you in that research. So, and this is the funding agencies, and I would like to thank you for your attention.
We thank you for your, firstly, lovely messages, and after that, your contribution. If you have any question, we can continue with the questions. My friends help you. Yes.
Hello, Professor. Thank you for your presentation. For thin layer coating of the microorganisms, do you use chemical methods or physical methods? Coating part. Solid.
No, it's just entrapment.
Okay.
So, and we use also 3D printing when you have a bio-ink mixture of the, for example, alginate with the cell, and then by putting the calcium, with the calcium you can cross-link it, or photocurable other cross-linkers. So we are using different cross-linkers.
Thank you.
We have one more question here.
Hey. Hello,
Hey.
Yagut Hocam. Thank you for the very informative presentation. You mentioned about that solid state versus suspended, and yes, solid state is more algal density. That it has more algal density. But how about aerial production? For example, per meter square, is it still like more than the other one?
Yeah, volumetric production is definitely high, but aerial, of course, there is a problem, because if you have a really dense cultures, you need a thin layer and much larger areas, and that's the problematic part.
Ah, okay, thank you.
Gamze.
Thank you so much for the great talk. My question is, how significant the role of the NADPH for the hydrogen production section? The reason I'm asking is, you mentioned sulfur deprivation helps overall nutrient stress by sulfur helps hydrogen production. In green microalgae, not cyanobacteria, in green microalgae, we see major nitrogen limitation actually positively impact NADPH expression, more pyruvates, more bioproducts. So that's why actually there are more lipids in most green microalgae. How significant the role of NADPH in the biohydrogen production? You mentioned it has a positive effect, but how significant it is? If so, maybe other nutrient limitation might also positively help.
Yes, I mean, we are mostly trying to go through direct water photolysis, which means that we are using ferredoxin as a donor to hydrogenase. So in this case, actually NADPH doesn't really play a role as a lot in our system, in our protocol. But what you mentioned, it's related to nutrient deprivation, because in this case you have indirect pathway when there is a photosynthesis and you have a CO2 fixation, starch accumulation, and starch degradation. And that's why NADPH plays quite a big role there in nutrient deprivation.
Then thank you again for your valuable contribution.